Hello, my name is Daniel Sandberg, and it's my great privilege to introduce an undergraduate research project from the biology department here at Flathead Valley Community College, antibiotic potential of lichens, fungi, and bryophytes in Northwest Montana. This all began as a question from one intrigued student and has since blossomed into the project you see before you today. Building on more than 6 years of research, we have tested over 40 extracts and have completed literally hundreds of experiments in the past year, all testing for antimicrobial potential of lichens, fungi, and bryophytes sourced from the greater Flathead Valley in Northwest Montana. The question of whether a local vegetation could potentially be used as antibiotics was asked out of necessity, as antibiotic resistance has become a real threat to global health. According to the CDC in 2019, more than 2.8 million antibiotic-resistant infections happen each year, which results in more than 35,000 deaths. Both of those figures are from the US alone. Finding new sources of antibiotics is a pressing need.
Hi, my name is Angela Wyzorek, and I'll be discussing our disc diffusion methods. So antibiotic activity was analyzed by a modified Kirby-Bauer disc diffusion method. The bacteria was inoculated in Mueller-Hinton broth for 24 hours prior to disc diffusion. Selected extracts were resuspended with dimethyl sulfoxide, DMSO, vortex and centrifuge to increase solvation and to settle debris. We pipetted the extract, positive control and negative control, onto the 16 millimeter diffusion disc. The disc were then placed on the plated bacteria and incubated at ideal temps for 24 hours. Zones of inhibition were then measured.
Hi, I'm Keith Moore, and I'll be going over our results. Figure A, the pie chart, diversifies the extracts which exhibited antimicrobial activity by type, with an overwhelming majority of positive extracts coming from lichens at 46% and the minority coming from fungi at 3.2%. Figure B is a display of our most positive result-producing extracts. This table demonstrates the inhibition of the test organisms against 11 different extract species. An X indicates there was a zone of inhibition identified when the extract sample was applied to an inoculated plate. Of these extracts, Bacillus cereus and Staphylococcus epidermidis were observed to be inhibited against all, whereas E. coli and Candida albicans were only inhibited by 3 extracts. Figure C, the bar graph, is a visualization of inhibition against the 6 bacteria and the yeast Candida. The positive controls are represented in green. Amanita muscaria, the fly agaric mushroom, is shown in yellow. Hypericum perforatum, or St. John's wort, is seen in red. And lastly, Ceanothus velutinus, a type of buckbrush, is charted in blue. It is important to note that the magnitude of inhibition does not necessarily correlate to the potency of the extract. The ability for active compounds in the extract to diffuse may have a greater impact on this observed inhibition.
Hello, my name is Haley Fennell, and I will be discussing the further research. An additional goal of this project is to begin isolation and identification of the active ingredients responsible for antibacterial activity. Over this past year, over 40 extracts have been tested for antimicrobial activity. Of these, Ceanothus, Hypericum, and Amanita have been the most promising. These extracts have consistently shown activity against multiple bacteria. The 3 extracts have been used in ethnobotany for many years, and there is minimal antimicrobial studies published on the extracts. Out of the published studies, these species have illustrated antimicrobial activity against Staph aureus. This coincides with the data collected thus far. Ceanothus, Hypericum, and Amanita have also shown antimicrobial activity against Bacillus cereus and Staph epidermidis. Moving forward with the project, the next step is to perform minimal inhibitory concentrations and begin isolation and identification of active ingredients responsible for antibacterial activity. Methods such as gas chromatography and mass spectrometry would be used to isolate and identify the antimicrobial activity compounds that represent the next step towards the development of novel antibacterial compounds to treat bacterial infections. For more information, go to our co-presenters, Ethan Harbaugh, Kate Smith, Terry Reidendower, booth number 5630 on Tuesday from 5 to 6 p.m. Thank you.